Okay, well, it's just after 11, and Goudarz, I know you said you have a longer talk for us today, so why don't we go ahead and get started? Welcome, everyone. Um, Dr. Molaei is a research scientist and medical entomologist investigating the role of mosquitoes and ticks in the transmission of vector-borne diseases. He works at the Connecticut Ag Station as the director of the Tick and Tick-Borne Disease Surveillance Program, and he's an associate clinical professor at the Yale School of Public Health. He has published numerous articles on the ecology and biology of mosquito and tick vectors and on their respective roles in the enzootic and epidemic transmission of arboviruses, as well as tick-associated diseases. Like I mentioned, he has a longer talk today, so if we go over time and you need to leave, feel free to do so. If you want to stay and ask questions, feel free to stay or drop them in the chat. This is always going to be recorded as usual, so you'll be able to review it later. Okay, thank you and welcome, Goudarz.
Thank you very much. Hello, everyone. Good morning. It's a pleasure for me to be with you today. I have some slides and I would like to go over those briefly with you. So in the interest of time, as it was mentioned, my research has focused on better understanding of interactions among vectors, host, and pathogen, and also investigating ecoepidemiology of both mosquito and tick-borne diseases. By way of brief introduction, we have up to 10 arboviruses in the region. 6 of them are of human health concern. Today, I will briefly mention one research project on Eastern equine encephalitis virus and leave the rest of the research programs that I have or we have on mosquitoes for a later time. Both West Nile and Eastern equine encephalitis virus, as you know, circulates in enzootic cycle involving ornithophilic mosquitoes and wild passeriformis birds as reservoir amplification host. And towards the end of the season, we have epidemic or epizootic transmission with the aid of so-called bridge vector mosquitoes. Our objectives have been to identify factors that facilitate enzootic amplification of West Nile and Eastern equine encephalitis viruses by quantifying mosquito populations and evaluating its relationship to virus activity, identify avian hosts that serve as reservoir or amplification hosts, assess the risk of human infection with these two viruses in different ecological settings by quantifying temporal abundance of potential bridge vectors and investigating their blood feeding pattern and infection status. And finally, examining land use characteristics and the spatial distribution of vector mosquitoes and human populations. As most of you know, Eastern equine encephalitis virus is the most pathogenic arbovirus in North America, and it leads to 8 to 10 human cases per year, 40% case mortality, neurologic impairment in those who have survived the clinical symptoms, and there is no commercial vaccine for humans, even though we have vaccine for equines. Activity has been in and around the freshwater hardwood swamps, and as I mentioned, the virus circulates or perpetuates in an enzootic cycle that involves wild passeriformis birds and ornithophilic mosquitoes. Prior to 2003, we used to have rather intermittent activity of Tripoli virus every 3 to 4 years. But since 2003, we have witnessed annual recurrence of Tripoli virus throughout the eastern United States, with the highest number of Tripoli virus human cases was in 2019, and in 2024, We also have had extreme triple E virus activity in mosquitoes, but thanks goodness it didn't lead to that many human disease cases. And majority of these cases occur in upper Midwestern states such as Michigan and Northeast, New Hampshire, Massachusetts, and then further south in North Carolina, Alabama, Georgia, and Florida. Over the years, we have investigated vector-host interactions of mosquitoes in triple E virus foci in Connecticut, Vermont, Massachusetts, New York, and in Virginia. We have identified a number of bird species in our studies as the most frequent hosts for Culicita melanura. Among them are wood thrush, American robin, tufted titmouse, and the list goes on. Similarly, in New York, we identified wood thrush, American robin, song sparrow, ovenbird, and other bird species. In Massachusetts, American robin was the most frequent host of Culicita melanura, and then tufted titmouse and black-capped chickadee. As you noticed, these birds are repeated. And in Vermont, interestingly, we saw green herons as the most frequent source of blood milk or for Culicita melanura. It's important to note that majority of these mosquitoes in Vermont were collected from a rather coastal region. That could be the one reason for this occurrence. We and others have looked at the antibody rate in some of these bird species that we have identified as the source of blood meal. As you can see, wood thrush, American robin, ovenbird, and some other bird species have had high rate of antibody for triple E virus. By looking at the mosquito abundance and host choice and a number of ecological factors, we we did some statistical and mathematical modeling, and we came across some of the bird species that serve as super spreader of triple E virus throughout the region. So in addition to melanura that we investigated rather extensively, and we found out that this mosquito is principally an ornithophilic mosquito with little inclination to feed on mammalian hosts. We also looked at Cochleatridia perturbans, Aedes canadensis, and Salinarius. As you can see, perturbans is an opportunistic feeder, and I know my colleagues in Massachusetts, they feel that this mosquito species is important for epidemic transmission of triple E virus, and I tend to agree with them. Then canadensis, less so, because it does appear that it has very limited interaction with bird species, and Salinarius, as you know, is the poster child of an opportunistic mosquito species. We also identified human-derived blood meals in these mosquito species. Earlier, you noticed that we do see some differences in blood feeding of melanura. That could be in part due to the host availability and abundance, but early on we investigated the blood feeding of Culex pipiens in relation to West Nile virus transmission, and we were able to demonstrate that Culex pipiens population, there is a hybridization or degrees of ancestry in Culex pipiens population that derives the feeding, one of the drivers of feeding on mammalian hosts. And we took a similar approach this time. Instead of using microsatellite analysis, we did high throughput genomic studies. And interesting, I'm drawing your attention to the top panel where you see K2. I don't know whether you can see my mouse moving around. So as you can see in New Hampshire, we did see some population structuring of Melanura, but we do not know exactly why we have this one and where that population structuring coming from or differences coming from, because in the case of Culex pipiens, we knew that we are dealing with Culex pipiens, Culex Culex molestus, and also Culex quinquefasciatus as the source of hybridization or admixture. Still, we do not know exactly what is happening with the population of Melanura in New Hampshire. We looked for 20 years when we were working on this manuscript. We saw that 60% of EEE virus positive mosquitoes belong to Melanura. Then other mosquito species we recently looked at, similarly, we saw that at this time even 70% of Eastern equine encephalitis virus positive mosquitoes are Culecita melanura. So one other investigations that we have done in our group to look at the EEE virus titer in field collected mosquitoes. Dr. Armstrong and Dr. Andriadis examined this one, and they found out that Culecita melanura is the only mosquito species that high titer enough for maintaining Eastern equine encephalitis virus transmission. So to wrap it up, we can conclude that the enzootic cycle of EEE virus is maintained with the aid of Culecita melanura, and to lesser extent Culecita morsitans in some areas and these mosquitoes transmit the virus or exchange the virus with passeriform birds, particularly American robin and wood thrush. And with regards to epizootic transmission, we have bridge vector. You realize that I have listed Culecita melanura as a bridge vector because the number of human disease cases are not that high. I believe that even 2% of mammalian feeding by melanura in some areas is sufficient enough to maintain this transmission to humans. And I have evidence that this is the case, most likely is the case, because melanura, we do see in some states like Massachusetts every year human activity of EEE virus, but in some states like Connecticut, we have rarely seen except for a few cases in the past few years. So therefore, melanura in some states could be a good bridge vector along with other mosquito species. So we have recently started a xenosurveillance project to better understand ecoepidemiology of EEE virus in the Northeast. Our aim is to characterize the source and factor mediating EEE virus emergence in the Northeast and elucidate the factors contributing to this sporadic occurrence of EEE virus outbreak. I would like to mention that although we have a relatively good understanding of EEE virus in the area, but there are some outstanding questions that we would like to address. The first thing is that we still don't know how the virus overwinters in our region. We have evidence for both occurrences. We have evidence that EEE virus is introduced on an annual basis to our region from the south. And we also have some kind of recycling of EEE virus locally, but we don't have exact evidence of how the virus overwinters. With this project, we are we are collecting mosquitoes from EEE virus site. We are testing head and thorax for EEE virus separately. We are testing legs and wings separately to determine whether there is disseminated infection with the dissecting midgut and splitting that in 3 pieces. One piece, we are doing blood meal analysis to determine the bird. We are using 1/3 to look at the antibody rate by ELISA or other tests to determine the exposure of the bird from which the blood has come to EEE virus. And then the last third of the blood meal is used to use PCR for determining whether the bird at the time that mosquito fed upon was infectious, actually. So I want to end the mosquito efforts, even though we are doing a number of other mosquito-related projects in our group, so and in my team as well. Now, I would like to discuss some of the projects that we are doing on ticks. As you know, ticks are becoming a major problem, major challenges, particularly in states such as Connecticut, that is at the forefront of the range expansion of at least 3 important invasive or non-native tick species in the region. As you know, tick-borne diseases has been on the rise just between 2019 to 2022. Based on CDC reports, we have seen 40% increase for a total of 180 some thousand tick-borne diseases in these four years. However, as you know, the actual number is far more greater than the numbers that are reported. So to deal or to respond properly to the problems with tick and tick-borne pathogens, we have 2 surveillance programs in our state. Active tick surveillance that was established in 2019 with the generous support from CDC. We are still conducting surveys in 40 sites in all 8 counties. This program provides information on the presence, abundance, distribution of native and invasive tick species and associated pathogens. Since 2019, we have collected and tested 20,000 tick specimens and reported the findings to the public as well as to ARBONET. Passive tick surveillance, also known as tick testing laboratory in Connecticut, was established in 1990. It provides statewide service to Connecticut residents and inform them of tick-borne diseases. So far, in recent years, we have been receiving up to 6,000 ticks per year. We also conduct targeted surveillance to investigate range expansion of native and invasive ticks and dynamics of tick-borne disease activity as informed by our passive tick surveillance. And so far, greater than 150,000 submissions have been received and processed to date. As I mentioned, we have been investigating the different tick species in our region. We recently, with generous help from Becky and Lars Eisens, we published this one in the Journal of Infectious Diseases. Over there, we expressed our understanding of the range expansion of both native and invasive tick species in the region, with a focus in Connecticut as well. In 2024, as an example, we received over 4,200 submissions to our passive surveillance. 80 some percentage of these are black-legged tick, about 13% dog tick, and nearly 5% American, Lone Star tick. And with the infection rate in black-legged tick ranges from 27,000 to 38,000 for black-legged tick with Borrelia burgdorferi. Then we have about 5% with Anaplasma phagocytophilum. It's important to note that we are seeing a rather increase or substantial increase in Babesia microti infection in black-legged tick. And then we are seeing about 2% with Borrelia miyamotoi and Powassan virus 0.4%. And then we have up to 6% of ticks to be infected with two or more than two pathogens simultaneously. With regards to phenology of ticks that are submitted to our laboratory, that also corresponds to what we are seeing in real field. We don't receive, understandably, that many larvae. However, submission of nymphs reaches a peak in the month of June, and I will explain this a little further. Have adult black-legged tick that reaches peak activity in the month of around May and later in November. If you are interested to find out what are the age groups that are exposed to tick bites, I would say that less than 10 years old are exposed to greater tick bites, and then people at the retirement age, and then those younger generation or teenagers or young adults, they would rather to sit at home and play computer games, and they don't have much to do with field activity. Good for them. So historically and understandably, greater submission of ticks in Connecticut are coming from Fairfield and New Haven counties. There is no surprise in that, because these are most populated counties in our state. However, we are seeing relatively fewer number of ticks coming from rural counties that cannot be explained just based on the population of these counties. In other words, it is under these counties are under, they are not actively participating in submitting ticks. This could be partly due to desensitization of the people. We decided last year to investigate barriers to engagement with our passive tick surveillance program in Connecticut, in collaboration with some faculty and MPH students from Yale School of Public Health. And I'm pleased to inform you that the result of this study is currently being written, and soon we will be able to submit this manuscript so that you could see what are the reasons for participation and or not participating in our passive tick surveillance. A few years ago, again with generous support from Lars and Becky, we looked at our historic passive surveillance. We realized that the nymphal black-legged ticks infection prevalence is highest in our rural counties, despite the fact that ticks are submitted from Fairfield County and New Haven County at a higher number. And it seems that this corresponds with the number of Lyme disease and other tick-borne disease cases in our rural counties. Again, Lars and Becky supported us to compare our passive and active tick surveillance. Our goal was really to better understand which one of these programs is serving public better, and whether there is a possibility that we could combine these programs should our funding goes further down and we can't sustain running our active tick surveillance, whether we can do targeted surveillance informed by our passive surveillance. And the finding of this program was that these two, active and passive tick surveillance, are complementary. However, it is a little bit, perhaps for me, premature to say whether we can drop active tick surveillance. But if we have no choice to drop active tick surveillance, based on our passive tick surveillance activity and informed surveys that we are doing in field by our passive tick surveillance team, we have been able to pretty much identify risk, identify invasive tick species in our region, and also important tick-borne diseases. So we are currently doing a research project with Yale and UConn based on the IRB that we have received to better understand the outcome of patients that have been bitten by ticks and they have submitted to those ticks to our laboratory. We are trying to determine if prophylaxis treatment has been prescribed. If subject who submitted ticks, they developed signs and symptoms of Borrelia burgdorferi, Anaplasma, and other tick-borne pathogens. If they were reassured versus still seeking medical treatment, if the subject received antibiotics or antiparasitic therapy without any sign of symptom, and if the subject received antibiotic antiparasitic therapy without any sign of symptoms of tick-borne illnesses. The goal of this project is to see whether we can come up with a solid science-based information so that we could share this information with public health authorities and CDC to let them know maybe we could come up with a set of guidelines with our physicians and those who are at forefront of treating these tick-borne diseases. Maybe we could save time, we could save hassle, we could also reduce the number of people that are taking unnecessary antibiotics. And as you know, we have different types of schools when it comes to treating those who have been bitten by ticks, let alone those who have shown symptoms of tick-borne diseases. We are currently working on estimating the prevalence of Powassan virus in Ixodes scapularis and disease outcome, because it's still one of the questions that we would like to answer. Apart from determining the frequency of Powassan virus in ticks, we do not know what is the spectrum of illness in patients who are bitten by infected ticks with Powassan virus and other tick-borne diseases. When it comes to Powassan virus, we really don't have a good understanding of what, in a nutshell, what percentage of people who are bitten by ticks infected with Powassan virus, they really show symptoms. And we are doing this one. We have hired a phlebotomist company. We are collecting blood samples and so on. So at some point, maybe at a later time, I could explain further. So one of the issues that we are dealing, we have always tried to get better information or additional information on the ticks that are submitted to our laboratory. Recently, we were approached by a team from Quinnipiac University, and We have recently completed the first phase of this project using metagenomic and nanopore sequencing to determine that we can use this relatively inexpensive methodology, not only get the information on the infection status of tick species, but also get better information on what is happening in ticks. And as you can imagine, metagenomic is providing a lot more information than basic PCR that we can get it. So the second tick species that I would like to mention is the Lone Star tick, the work that we have been doing on this tick species. The first report of this tick activity was in our region was from Connecticut, apart from some population that has been around, local population that has been around in Rhode Island, Massachusetts, and some areas, but the real established population we reported in 2018 from Manresa Island. And this is the picture of the deer that perhaps succumbed to heavy infestation with Lone Star tick. And later on, we raised awareness about the range expansion of Lone Star tick in the northeastern United States. As you can see, as you realize, Amblyomma americanum or Lone Star tick is a very aggressive tick, and they are abundant right now in our region. And we have numerous individuals who are receiving a lot of tick bites. And the tick bite alone, without even being concerned about infection, can lead to high irritation, even though if there is no pathogen involved. This is a lady who walked into our laboratory a couple of years ago, and this was the result of receiving a few bites from larval or nymphal Lone Star tick. And this lady ended up going through antibiotic treatment for several months in order to clear all these rather debilitating allergic reaction. I don't want to bore you with the number of pathogens that Lone Star tick is involved in transmission, but I want to emphasize on the alpha-gal syndrome that was first reported in 2009 in the United States, and then the number of confirmed cases has increased to 34,000 by 2019. And we are currently working on a project to determine the frequency of alpha-gal syndrome with and without established population of Lone Star tick. As you know, our state is unique in a sense that we have establishment of Lone Star tick in our New Haven and Fairfield County, but we do not have established population in other sites, the other counties, and we want to see whether there's any difference in the frequency of alpha-gal syndrome in areas that we have we have Lone Star tick, to determine the relative frequency of alpha-gal syndrome, and also investigate the spectrum of symptoms and frequency of alpha-gal syndrome in patients. We have a manuscript right now under review. It's under revision. It has been accepted primarily, and we are reporting the first human disease case from Connecticut that we believe that a male Lone Star tick was involved, and this male Lone Star tick was infected with a unknown Ehrlichia species. We are trying to further characterize this Ehrlichia species, and also the tick was infected with Rickettsia amblyommatus. We first reported the established population of Gulf Coast tick in coastal region in our Fairfield County, and we published that in 2021. And this was the first time that we saw this tick population in the northeastern United States. Up to that point, the northernmost population of the tick species was in the state of Delaware, and we reported about 30% of specimen species to be infected with Rickettsia parkeri. And when at the time we submitted this manuscript to a different journal, we were told that you are not reporting human disease cases. We unsuccessfully argued that it's just a matter of time. It turned out just last year we had the first human disease case of Rickettsia parkeri rickettsioses in a 21-year-old female. And if it wasn't our intervention, I don't know what the outcome could have been, because we don't test Gulf Coast tick in our laboratory routinely. But this was a collaboration between our agency and health department that the lady, the patient lived in, and we were alerted that the patient is sick, and we immediately tested the tick, and it turns out to be infected. Then we dispatched efforts, and we met with the patients and with the physician, and with the involvement of CDC, we were able to document the first human disease case of Rickettsia parkeri rickettsioses in the northeastern United States. Longhorn tick, as you know, I don't want to bore you with the longhorn tick, but this tick has now established population in 21 states and District of Columbia, and it based on climatic condition, it is expected that this tick will take home in every state and perhaps Canada pretty soon. And this tick, as you know, has been reported to be infected with up to 30 different tick-borne pathogen in its native and invasive range. In the United States, Theileria orientalis has been documented and confirmed in this tick species and transmission as well. We and others have reported on the evidence of DNA or infection with Borrelia burgdorferi, Phagocytophilum, Anaplasma phagocytophilum, Babesia microti, and Bourbon virus. And it's also a vector of Rickettsia rickettsii. And it has been shown that this tick can transmit heartland virus. We recently reported about feeding in this tick species that highlights the fact that this tick can acquire infection from an infected host during the first blood meal, and when this tick attempts to bite a second host, even though if this tick is not be competent, which we don't know, it still can transmit pathogen to next host in an attempt to complete the blood meal. We recently reported finding of Ehrlichia chaffiensis DNA in Haemaphysalis longicornis for the first time in the United States. It is important to note one of the major concerns that we have here in our state is that populations of Lone Star, populations of longhorn tick and Lone Star tick are sympatric in coastal region in in Fairfield County and New Haven counties. And we do see now evidence of not only pathogen exchange in the case of Ehrlichia capensis, but others as well. As you know, white-tailed deer serve as a host for both lone star tick and longhorn tick, and also is a reservoir host for Ehrlichia capensis. And it seems that it is going to be a major public health concern, even if longhorn tick doesn't bite human, which it does, but not perhaps as frequently. It does appear that this tick is going to become another problem with tick-borne pathogens such as Ehrlichia capensis. The last tick I would like to briefly talk is the American dog tick, or Dermacentor variabilis. This tick species, as you all know, transmits rickettsia, the agent for Rocky Mountain spotted fever. First of all, I would like to mention a few things about this tick species. The population of this tick species in our state is increasing rather. Historically, we have had about 13% of this 13% of this tick species, but we are seeing now up to 23% of all ticks submitted to our laboratory to be American dog tick, and this pretty much reflects what is happening in the field. Another information or evidence that we have is that prior to 2016, we didn't have we didn't have any Rocky Mountain spotted fever cases in our state. However, since 2016, we are seeing every year up to 5 cases, sometimes more, Rocky Mountain spotted fever cases. And in the past couple of years, we have had mortality in our state. Last year, 1, and the year before also we had another case of mortalities. We have tested hundreds, perhaps thousands of American dog ticks. We have yet to come across a single American dog ticks to be infected with Rickettsia rickettsii. We have a hypothesis that some of the tick-borne diseases that have been diagnosed as Rocky Mountain spotted fever, result of Rickettsia rickettsii activity, may not be actually Rickettsia rickettsii. We have identified some other Rickettsia in our region that are transmitted by other tick species, such as Lone Star tick, and we believe some of the Rocky Mountain spotted fever cases, human disease cases, might not be the actual Rickettsia rickettsii diseases. So we are investigating that as well in our region. We are also looking at the feasibility of Malditov to resolve morphological and genetic similarities between in tick populations. We published this one with leadership of Dr. Chris Paddock at CDC. Now we are looking at the feasibility of Malditov for resolving situation with longhorn tick, and also finding a way to see whether we can use this methodology, this rather inexpensive methodology, for pathogen screening as well. In the past several years, we have reported a number of invasive tick species that have come to our state by human travelers, and we recently have a manuscript submitted, and in that we are reporting at least 7 invasive tick species that came to Connecticut or through Connecticut to the United States, including Amblyomma mixtum from Guatemala, Amblyomma mixtum from Costa Rica, Amblyomma coleps from Belize, Rhipicephalus pacellus from Tanzania, and then we had at least 7 specimens coming from Germany, Scotland, and Poland, and hopefully that will soon will be published. It has been accepted. It is under publication. So most of you are familiar with the factors that driving range expansion of tick and tick-borne diseases, including land use and land cover changes, habitat fragmentation, changes in vegetation structure in host community, increased diagnosis and reporting, and climate change. We recently, as you have noticed, we recently published a paper to further highlighting and emphasizing on the role of white-tailed deer as a reservoir and reproductive host, as well as dispersive host for tick and mosquito-borne diseases. And hopefully these initiatives would create some momentum to really find a solution to deal with deer population. I want to leave at that point because this tick has been controversial. This issue has been controversial, and in the past we took attempt to deal or to address or to find a solution, but we didn't have much success due to some political and non-political reasons. We are seeing increases in the population of all 3 tick species in our state with regards to black-legged tick and dog tick. We do see fluctuation of Lone Star tick is steadily increasing. We looked at the impact of climatic and environmental condition on the 2 tick species, black-legged tick with pervasive population and American Lone Star tick with recent established population in our region. We found out that it does appear that because black-legged tick has a rather pervasive population, it doesn't appear that at the local level, I emphasize on that, climate change or climatic condition has much impact on the population of this tick species, but clearly it is impacting the population of Lone Star tick. And as a result of these findings, we are currently investigating to determine if a warming environment alters the density of endogenous symbiotic bacteria in black-legged tick and Lone Star tick, and determine if the warming environment alters transmission dynamics of Borrelia burgdorferi and Ehrlichia chaffeensis with these tick species. This is a collaborative work between our laboratory as well as the Yale Dr. Errol Fickrich's group. So control and competition. So as Lars has contributed in our joint publication, and I support this notion that we will likely have better success for controlling ticks that have narrower host range, particularly domestic animals. But when it comes to those tick species such as longhorn tick that uses a number of or up to 50 or maybe more host species, it is a challenge. Still, we do not know whether there will be competition and if there is any competition between the tick species that we have in our state, and whether that competition will be host-based competition or whether there will be some environmental competition. You know that there are evidence of competition between Lone Star tick and American dark tick in Southeast, and also Lone Star tick and black-legged ticks in New Jersey and Connecticut. Finally, I would like to wrap it up by saying that vector-borne diseases will continue to be a major concern, and we will see additional vectors and pathogens emerging. We will see range expansion of both mosquitoes and ticks and associated pathogens in the region. We will see, I would like to add that transmission of some of the arboviruses depends on the frequent infection of mosquito species with key virus-competent bird species, some of which serve as super spreaders. I already highlighted some of these host species in case of Melanura. And finally, uncertainty in critical aspects of vector-borne diseases will hinder our efforts to accurately quantify and predict tick-borne disease and risk associated. And needless to say that there is still an urgent need for effective, widely accepted approaches to prevent vector-borne diseases. Thank you very much for your patience. I would be more than happy to answer if there is any question.
Thank you, Goudarz. Yes, if anyone has questions, we have a few minutes before noon, and we're happy to go over a few minutes as well if we're still talking by then.
Hi, I'll start off. Matt Cahalan from New Hampshire. I've been a PI on a CDC grant looking at climate change and impacts on public health. This is probably a question that might be shared by the presenter and Lars Eisen. I'm just really interested in learning more and understanding that kind of your perspective on this attribution evidence for kind of host versus land use versus climate. I know that Lars, you wrote an article or published a study, a review last year looking at climate change, or maybe it was 2023, and determined that really it's hard to tease it out based on the quality of the data. And I'm always interested, well, how much is the host versus normal climate variability versus anthropogenic climate change? So if you have any more insights on that, like where would we get to, how would we improve the data to be able to do that? That would be great to hear more about insights there.
Yeah, the information that I could share at this point are more like anecdotes. Unfortunately, I do not have a solid data to share with you, but based on just face value, what I am seeing, that it does appear that there is some competition, at least maybe competition is not a better term. We are seeing some of these invasive tick species in some counties in our state. It does appear that they are not bothered by other tick species. They coexist. But we are also seeing some tick species, notably they are absent in those areas. I can let you know, just for instance, black-legged tick in areas that we are seeing Lone Star tick and Gulf Coast tick, and to that matter, Asian longhorn tick, we don't see. It does appear that it is absent. That doesn't mean that the area is not suitable for black-legged tick. As you know, black-legged tick is pretty much flexible when it comes to the habitat, but we don't see. It does appear, at least that's how I understand, that we are seeing both at the environmental level and as well as host. But as you mentioned, testing that hypothesis, particularly when it comes to competition on host is a daunting task, which I don't have expertise to allude on that further. Lars, please feel free to chime in.
Yeah, I can pitch in a little bit. I think this started a bit with us thinking about the modeling that has to do with climate change, and I've been interested in climate change for a fair amount of time, going back all the back to stuff that I was involved in for Ixodes Ricinus in Sweden, where very clearly my grandparents had a farm. There were never any ticks. When I was in my mid-20s, the tick was coming in from the south, and it's been moving north in Sweden. So I'm very clear on that. But then there was also, in addition to the climate warming in Sweden, there was an explosion in the roe deer population. And it's a bit the same here. We have, at the same time as we have a climate warming over the last 40, 50 years, we've had an explosion in white-tailed deer. And it makes it hard to tease out which is which. And a lot of, based on the lack of data for host communities and deer, a lot of the modeling for climate change driven impacts on Ixodes capillaris and other tick species have just ignored the hosts. And if you have in parallel increasing temperatures and other meteorological parameters that favor the tick, as you have host communities, a key host improving, you will see strong associations with climate, but it's not necessarily the only driver, or maybe not even the sole driver. So that I think that's the point we were trying to make in our paper, that climate is important, but it's hard to tell how much of the change in the last couple of decades can be attributed to that versus the deer population increasing and spreading from refugia after having been virtually exterminated in the so I don't have a good answer for you. I think there needs to be studies that consider both, and maybe now that we're coming to a point where we have very high and stable deer populations in some areas, it would be easier to tease out into the future what the effects are of deer versus climate relative to an area where you have still an increase in the deer population, which has happened if you go back 10, 15, 20 years. So that's how I'm sort of thinking about it. So I think it's both, but it's not clear. And I think there was just too much emphasis for a little bit on just climate, and it was all climate. So that's where I am on that right now.
Thank you. That's a helpful insight. And I see the same thing in New Hampshire, that everybody wants to claim it's climate, but we need better information, and maybe that data will come out at some point when things stabilize. Thank you.
Since I was talking here, Goudarz, if you don't mind, how soon will the metagenomics pathogen detection come online? Because that will become really interesting in terms of you will start to find all kinds of other things that you haven't been testing for before. So
yeah, we are writing. That's a good question. We are writing the manuscript as we speak. We are meeting tomorrow to tackle some of the issues with writing. I'm hoping that within a month we will be able to submit it, and then, as you know, Lars, God knows when it will be available. We are hoping that we will have a relatively easy ride when it comes to publishing it. But I'm very optimistic that we should be able to submit within a month.
So that will include separating out the human and non-human variants of Anaplasma phagocytophilum, I assume.
Correct.
Yeah. So do you have a sense for that in general at this point? How how much of the Anaplasma phagocytophilum that you've been testing is the human variant versus the deer variant?
No, I don't. Right now, I don't have. Sorry,
Okay.
that inside. All right. I think that there are no additional questions, and I want to take this opportunity one more time to thank you all, and I wish you all the best and happy summer days. Okay.
Thank you for coming, everyone.